Hello, I'm going to try to give you a brief overview of our project on the biological control of wild ginger, namely Hedychium gardnerianum, or commonly known as Kahili ginger, which you can see here with me in the picture. So what are wild gingers? Well, they're very popular ornamental plants belonging to the Zingiberaceae family, and they're still widely sold all over the world today. There are many different species, all highly fragranced and different varieties, and you can even see some down the road in Savile Garden if you like. Here it is in Savile Garden. But as often happens with ornamentals traded the world over, some have escaped cultivation to become serious weeds, and this weed in particular, Kahili ginger, is listed as one of the world's worst 100 invaders by IUCN, and is the one I'll be talking about. So in its invasive range in Hawaii, as we see here, and New Zealand, for example, it forms dense monocultures in the forest understories. It's got incredibly stocky rhizomes, you can see here, and basically it prevents recruitment of natives and alters whole ecosystems by just encroaching on intact native forests, and it threatens rare and unique fauna and flora. So with such dense and often inaccessible populations, current control measures are proving completely inadequate, and here we see it in La Réunion in the Indian Ocean island. So in 2008, CABI were funded by a consortium from Hawaii and New Zealand to look into the potential for biological control of Hedychium gardnerianum. Where are they from? Well, Hedychium species are largely native to the northeastern Himalayas and southern China. So we visited herbaria in the UK to look at geographic records for the species of interest. And it's always encouraging to find evidence of insect or fungal damage when you're looking through the specimens. So we found that India, and in particular Sikkim state, looked like a good place to start looking. So to cut a long story short, we eventually signed an MOU with MBPGR in Delhi to facilitate this and the Himalayan Balsam project. And then we flew to India, to New Delhi, and subsequently on to Bagdogra in West Bengal. Okay, and then from there it's just a bumpy 4-hour drive to the hill town of Darjeeling, following up behind the toy train sometimes, and you can see the tracks there on the right hand side, up the windy roads with, there's the toy train, with the occasional stopover to do a bit of DIY maintenance on the engine, and then you arrive in the wonderful town of Darjeeling, which is bustling, and you get your luggage carried up to the hotel for you. It's also an excellent opportunity to look at the local botanical gardens and get your eye in for the plants, the Hedychium species, because there are so many in Sikkim, and have a look through their herbarium records too. And since you're in Darjeeling, it'd be rude not to have a cup of tea as well. So overnight in Darjeeling, and then it's on to Sikkim, which is north of West Bengal. Another 4-hour drive to get to Gangtok, which is the capital in the eastern district. And you have to go through Rangpo, which is a border patrol town, and get your permits to be able to stay in Sikkim for 14 days maximum. Sikkim is very famous for its amazing orchids and its rhododendrons, so it's a very beautiful place to survey. There are very few roads in Sikkim, and they're fairly treacherous to drive. We certainly wouldn't attempt it ourselves, so this is Raj, our driver. Although we have government level collaboration, we still need to have state permits to collect material, and often we have to be accompanied by forestry officials to even go into forests. So then the survey can begin proper, and it's mainly driving around, trying to spot the plants, and often they're growing in fairly inaccessible places. But this is the native range, so here you're hoping to see the plant damaged by natural enemies such as fungi and insects. So we started in 2009. It's important to visit at different times of the year to capture as many suites of natural enemies as possible. And it's a team effort. We've benefited from lots of CABI colleagues going and collaborations in Sikkim, as well as training up some local students to help with the project in the future. So what have we found? Well, thankfully the plant in the native range is under severe natural enemy pressure. So we found leaf spots and rusts, as well as Lepidopteran larvae, stem borers, weevils, And we're still finding a lot of new things and consolidating the inventory. And we look at all stages of the plant, the seeds and the rhizomes as well. And because it's closely related to edible ginger, it's also important to have a look at those crops when you see them in the field, just to have a look to see whether it shares some of the same natural enemies. It's also really important to take lots of photos, since until this year we weren't able to export any specimens. And also important to talk to villagers and get local names for plants. People tend to be very knowledgeable, certainly about where plants are, and often they want to lend a hand looking for it or collecting insects, and even get their picture taken with you, usually when you're looking field weary and slightly bedraggled. But it's really good to talk to everyone you meet. So this year we were very lucky to get permission to survey in the Katsanzunga National Park, which involved a nice long trek. And the forest is protected here, so it's the most natural and unspoilt area for wild gingers. So here we are, we found the gingers, and we were looking for a fly that we'd found previously, which is really interesting, which stunts the plants by mining in the stems and prevents them from flowering. So what we usually do is find a plant with the characteristic symptoms and cut it open to see if we can find larvae.
Look inside and you can see it's all black and mushy, so something's been boring inside and all the way up the stem and it's stopping the plant flowering.
So there's the symptoms that you see in the field. The blackened tip is characteristic, and then when you cut it open, usually you find the inside is all black and mushy, and usually you have either pupae or larvae of the fly that we're looking for inside. So this is the larva, for example, and it's got this long tail to help it grip on the inside. The pupa are also distinct, and eventually the adults emerge into this fly, which is a chloropid. At the moment, that's the agent that we're looking at. There's a whole fly complex associated with the plant, though, and we're still looking at the biology of all of these, now in quarantine in the UK since we finally got export. So then after a few spins on the prayer wheel and a bit of sampling of the local hooch, it's back down the mountain to the airport at Bagdogra with all the specimens in tow, and a flight back to Delhi, where our collaborators at NBPGR are. There we deposit lots of specimens. There's a whole team of people. Rob was there for a while, but Jyoti works on the specimens that we leave behind. And then it's back to CABI to begin host range testing on the plants we've been growing in propagation. So I hope this gave you an insight into a typical field trip. It's not all glamour and G&Ts, but you do get to see some remarkable sights. And the red tape is a little bit tangled at times, but there's some amazing wildlife, and after a while it does start to feel a bit like home. But it's certainly always incredible India.